my graphs?
It's a great question.
If you go to Safari,
Zoom in on Hercules a
Chrome,
little.
sorry. And there's science dive.
That's good. Pull back a little
s-
tiny bit. Yeah, perfect. Thanks.
Definitely suggest being enthusiastic about nature and exploration.
I would add be yourself. I think the crew here
Okay, come
is
wide a little bit.
very diverse group.
Coming down
Amazing
some more, too.
backgrounds.
I'm giving you a pull.
Where am I seeing this temperature readout? Where am I seeing this temperature readout?
Is that 10.4 is not here?
on
Is
the
this an old
sides.
maximum, is it?
Yeah, it's an old max. It's
Oh, this guy?
2.6 right now.
Oh,
Is there a way to
2.1
reset that?
and 10.
Yeah, there's if you take that trackpad,
Yep.
and then if you see that little horizontal bar there.
Oh, the one that says reset max.
Yep, that one.
Perfect.
Should work.
So it's 2.8 now?
It's crawling a little. But it's
There we
pretty,
go.
it's pretty low. We should maybe wait for another 30 seconds to see what happens.
Nice. Yeah.
Do you guys see that temp readout? We have it up on there as well.
Oh, great. Thank you.
Yeah, here. So it's what, up to like 3.1?
It's pretty low,
Yeah.
low shimmer, so that
Yeah.
a little warm.
Some of you may notice we have a couple of hitchhikers on board, both alive and hanging on. Been a few attempts to try and remove them from the ROV, but we want to do it gently, so just hanging out.
Oh, there's a little jump.
Hmm?
Temperature keeps going up,
Yeah.
but it's a very slow pace.
Okay. Should we look at another spot? Yeah. Let's try and find another location maybe where we
Sure.
can see a shimmer. Do you want me to log this still? What we
Yeah,
found
yeah.
here? Yeah.
Because it's above background and there was shimmering there and
Yeah.
low. Because spots have low venting now doesn't mean that when they come back again, it won't be higher
Right.
venting.
There's some, it's pretty much out of our reach.
To your
I feel like I see shimmer almost dead ahead, but
Yeah, kind of to the left a little bit.
Yeah.
Aaron, is there a
Well, I see
label
it.
for this marker? Like
I see-
any?
what was that?
Is
do,
there
do you see
a marker
the,
here?
the kind of line of peach anemones?
Yep.
So it's definitely shimmering over there as
Yeah.
well.
Yeah, I can try and scoot up there.
Yeah, if you can, that would be great.
So you guys are talking about here, yeah? With the pointer stick?
Oh yeah, that's exactly where I
Okay.
was talking, Aaron. Thanks.
Yep.
So to the upper right of our screen, there's kind of a line of these peachy anemones with some octopus interspersed. And if you look at them, it looks like they're in a dream sequence of a movie. It's like,
I don't know if we'll have enough scope for
and
that.
What does
over
that
there.
mean?
you know when they go into a dream sequence of a movie,
Oh,
and
okay.
they shimmer the
Yeah,
picture.
I follow now, sorry.
Like you're going to sleep or going into a dream. It kind of looks like that.
I don't know if we'll have enough scope still to get over there.
There's a shimmering spot just to the
Oh.
left of us too.
In front of the octopus that's upright.
Upright octopus.
So there's an octopus that's looking at us, and
Yeah.
it's upright, and it's moving our
So-
way. There's a shimmer
There?
right at the tip of its longest, the arm that's
Yeah.
outreaching.
And just above the lasers.
Yeah. So is
what
that
I
one
can
that
do.
we could reach?
Maybe. We're very stretched out here,
Okay.
and that's
Or-
far away for us to get without sitting on anything.
Any of these kind of open holes right in front of the basket too. I mean,
You want to try another 10-meter move in?
Sure,
Yeah, I think
will
so.
do. Same bearing, 300.
Or 290 is spot on right now.
Okay. Bridge nav. Can I have a 10-meter step bearing 290? Thank you.
So Amanda is asking us whether or not this one that's out and moving about is a male. when we zoom in, we can take a look at the morphology of the arms as well as.
Go ahead and zoom video.
No.
Now this is a female. We're looking at the tips of the arms and they're all similar. So I would say this is a female. All right. There's a lot of shimmering here, especially to the lower left. There's something, a little crevice over there we could put the probe into.
Okay, full wide.
Guys, we've got over 300 visitors watching online right now from almost 20 different countries around the world.
Well, welcome to our day of exploration and this feature by the Davidson Seamount in Monterey Bay National Marine Sanctuary. We're excited to have you with us.
Jennifer, I think it was you were talking about, we were just looking
Zoom
at
in there video.
the one octopus there, and you mentioned that you thought we were looking at a female.
Mm-hmm.
How do we know the difference between a female and a male moose octopus?
Well, if you were to take that octopus that's out and think of her, if you were kind of looking down on top of the
Okay,
octopus,
full wide for a minute.
and her eyes were pointed to the top,
you can zoom
and
in
you
again.
turned her into a clock, you'd start counting her arms, moving clockwise. The first arm to the right would be 1, and if you count over to 3, in a male, that arm will have a modified end to it called a heterocotylis. And it's shorter and wider and doesn't have the suckers going all the way to the end. And so that's how we can differentiate a male from a female.
What's that little blue thing in the crack there?
I think that's one of those greenish, bluish worms, but I'm not certain, that we were seeing earlier.
Oh,
Okay.
one of the spoon worms?
Yeah.
Okay.
You guys want to hold it here or sniff
Yeah,
around
where
for
the temperature's increasing and
Okay.
the probe tends to respond relatively.
Do you want me to reset
Casually.
that max every time we pull the probe?
What does that exactly do? What is this max?
It just clears where it says max temp and it'll start building back up again.
So the green line there, she could reset it so that we would just see it.
I just want to, yeah, as long as we're seeing the real temperature.
Okay, all right.
the top line.
Yeah, I'll
Okay.
probably look at the graph.
Okay.
So this one isn't so far as warm. Is there possible to push further in?
I'm in there as best it can be.
Okay.
That's what I was saying. I can move around to different places.
Yeah, let's try just poking around to a few other places.
I'll try what I just moved it to for a second. See if that does anything. It's going down. You can see some shimmering just under it, just in between the coils there.
Mm-hmm.
Could try that out.
See, we've got a couple of reefers listening in right now from the Marine Science Institute. They are interns at the reef, which is the aquarium at UCSB. We've got Iris, Nelson, and Katie R. Hey, guys.
That didn't seem to get warmer, did it?
No.
Let's,
Well, we are in one general vicinity. This water might all be coming from the same ultimate conduit.
Yeah.
Because this is a big pile of rubble.
Want to pull
So-
out wide and see if we see something?
Yeah, it might be worth if we look over to the left. I don't know if the camera has that capability. There was some shimmering up on that as you moved up the kind of vertically up that wall. That I'm not seeing it now.
Want to zoom in a little video? Open the iris a little bit.
well, lower left, but
Yeah, it's not so obvious
Yeah.
to me now.
Yeah, 10 meters.
Can we, or do you want to take any other measurements that we can reach from here and then reassess? Or?
I mean, we could definitely stick in another hole towards the left where we haven't done that yet, but all the flow looks kind of evenly shimmering.
Full wide video.
Okay.
Aaron, is there a marker number for this site?
Okay, go in again.
We're just calling it Octopus Garden in general.
That's
Okay.
good there. Yeah, I guess there's some more, be a bit of a reach. Some more shimmering stuff right to the center. That's close to where I was though.
Yeah.
Yeah, we may have to reposition to try to measure. You wanted to go towards the left?
Yeah, well, when we were in the original position, when we first landed, it seemed like up as that wall kind of created itself to the left, there was a little bit of shimmering near a brooder in there. And I just suggest that because it's away from this general rubble pile, and so maybe it is a little different, maybe not.
Okay. Full wide, please.
And we're getting a lot of questions about why the octopus are at this place, including one from some marine science students in Kluben Biological Station in Sweden. Is there a reason that these octopuses are all around the sea anemones?
That's a good question. I do not know the answer. At the other site with the octopuses in the Pacific Ocean, which is 2,000 miles from where we are now, we don't see the same community structure. So this exposure at Davidson is the first I've seen of this pairing between the octopuses and the anemones. But that's definitely something that hopefully we'll be able to learn from the data that we collect on these dives over this trip, and then previous dives to the site and in the future as well. Good question.
Yeah, while this is not a new discovery, it's still a very early discovery with these octopus and trying to characterize the sites that they're at. And the questions you guys are asking are great questions. They're questions that our scientists are currently asking, and we're going to try and learn more about these octopus in these environments.
Do you see the big brazingid that's to like at 11 o'clock up on the wall?
On the wall, yeah.
Back there we see shimmering. I'm just curious, do you think you could perch on that kind of flat area where there aren't any octopus?
Maybe. Let's see. Might not look good though. That's pretty steep there. We might get the might not be able to get the bumper bar in there. I don't think.
Screen this. This is a really neat kind of crack feature we got going on though.
Yeah, I don't think
maybe could we follow this up just a little bit?
Sure thing.
That move settled out. You want to bring it in a little more?
We can see. It looks like it'll probably settle all right. If we could go back down there
and kind of see where this goes.
Leave it for another couple minutes.
Okay.
Unless they want to hop into that little canyon. Science, do you know, we still have the PCB pump running. Is that?
That's fine. If we think we're going to kick up sediment, we should turn it off.
But okay, but it's okay to have it keep going.
Yeah, so far so good.
Roger. Thank you.
Thanks for asking.
You want to push forward in there?
If you can fit, otherwise just maybe like looking to the left or the right. The kind of crack kind of seems to split here.
Yeah, that's right. I can come in here a little bit. I can.
Yeah.
Okay, maybe we get a little closer and we push them with video and see how much shimmer we can.
There's a little track of them running up here.
Yeah.
Okay, I'll look back here first. Do like a partial zoom there, video.
Kind of curious about what this canyon is.
It's good.
This is just amazing. For those of you guys watching, this is our last expedition for this year. The ship is going to head down to San Pedro to dock and then down to Ensenada for a brand new shiny engine. We'll be out again next year. We're getting a lot of questions about the Octopus
Down
and their
there.
position. Why
Looks
are
like
they
a senescent
upside
one.
down?
Stereo. Do you see that one octopus down in the canyon there? Looks really senescent.
Can we zoom in on that?
Yeah, it's
This
kind
one
of in the center of the lower part
Yeah,
of the screen.
the
Yeah,
bears?
just
Right
below
there.
the lasers, that's right.
Yeah, zoom right in. It won't look the greatest. But
Yeah.
Wow.
Okay, so
Yeah.
there is one that's done. Well, maybe I still see a little bit of respiration that happened, but that animal's dying. You think the sea star is going to, so Amanda just popped in, she thinks the sea stars are descending to start consuming her.
I wonder how long that takes them.
Right, and then, yeah, I think that's what another one of those brittle stars there, though, that was taunting people earlier. So the odds are that, you know, she's not here
still
because
breathing?
of shimmering, but she is still breathing, huh? Just barely. So for these females, when they're done brooding, this is what's going to be happening with them. They've used up all their energy, and they will be dying and then becoming part of the local food web.
See, is that shimmering that is kind of to our left, or is that just my eyes playing tricks on me? If we were to poke this wall, kind of right below the arm tips of the...
Of the octopus?
Yeah.
I don't think we can actually get in there. You want to pull back
Okay.
up
Oh yeah,
wide
we're
on
zoomed
the
in. I'm
camera?
sorry.
Yeah, we're zoomed in pretty far.
Forget where. This is a canyon.
But yeah.
We've
Full
got
wide.
an explorer who's 4 years old that would like us to know that there is no need to water the plants down
Okay, full
here.
wide view.
No, there isn't.
Yeah, I want to go sit in there.
Oh, I love it with 4-year-old explorer. That's exciting for us to know. We've got you watching with us.
Starting them young.
Look at all those octopuses in that crevice behind that, kind of protected in that rock area over there. I'm referring to the right side of your screen for people wondering what I'm talking about.
Oh, look at those tucked in down there.
Yeah.
That rock is very dark.
Yeah, that rock looks quite black over there.
Is that an open enough area that we could get a little closer and look to
Yeah,
see
we
if they're
can sit
shimmering
over
down
there,
there?
I think so.
Great.
We just come up a bit and see what's on top of it.
And then also there looks like there's
Oh,
a
yeah.
pocket on the face in our lower right screen.
Take a peek at that first.
Jennifer,
Yes.
octopuses, octopodes, or octopi? Tomato, tomato.
Oh, goodness. I've seen a couple different things on this. Octopuses is definitely a term that is accepted. I have heard of octopods, but that's more talking about octopuses as like a grouping instead of individuals of the same species. Any other?
I have a strong opinion that octopi is not correct.
Octopi is not correct, yes.
Yes.
So,
It's an old school
But there are
word.
two other options, octopuses and octopods. I mean, that's a lot of options to describe a group of
Yes.
an organism.
that's right.
We've got some 5th graders at Saunders that would like to know what type of octopuses we are looking at.
Oh, okay. Well, we are looking at a type of octopus in a group that's called the genus, which is the first part of their scientific name. That's Moose octopus, M-U-U-S octopus. We think it is the species Moose octopus robustus, but we are
Do a
having
little zoom there, video.
an octopus expert help to confirm that
Good.
that is the species that we are looking at here.
If we can land, it might be worth just sticking the probe in one of these places just to see what it is.
Yeah, sure. Okay, full wide.
So did you guys hear that we hit the record for sea temperature on the last dive?
Yes, 10.4. We
Yes.
haven't gotten anywhere close yet.
Right.
We're going to try and break that.
Yeah, okay.
Yeah.
Having a heat wave.
I'm kind of a competitive person, so I like to have these little goals to try to beat.
You betcha.
The difficulty here is finding a spot where we can safely set down
Yeah,
and
it's
reach where we want to put these instruments.
pretty steep. Yeah, the spa was perfect for that. Kind of flat.
So flat, yeah. So it's possible that if we can't find a spot that's really warm and pumping, we don't have to leave the temperature probe here. We're going to start after this surveying the greater area and looking for additional locations.
If you can get right in front of the lasers, that looks like it's a shimmering hole. I don't know if that's too far in front of you, though.
No, that can be done. You move the
Like a
bubble
little
cam up there,
above it where
Jess.
that hole is.
Thanks. Zoom in there, video. You need back here?
Yeah, right in there.
Okay,
Come
say
back a little bit
aw.
on the zoom. Perfect. Might not reach that. I don't know.
And if that's not going to work, there's a squat lobster on your right, just below the blue cord for your T-probe. That might be a good alternative.
I'm in there now.
All right, let's see.
That's pretty solid, though. It looks dark, but I'm up against a rock here now.
Okay.
I don't expect we'll see much out of that.
Not a
Yeah.
lot of shimmer there.
No, there's not. What about, down and to the right a little bit?
Okay. Okay, wide on the video. Down to the right. Oh yeah, okay. Zoom in there. That's good.
I'm not seeing as much shimmer here.
Yeah, I barely see any,
Yeah.
but it's curiosity.
Taking the temperature of the Earth.
Might have gone up a smidge.
Yeah, it's moving up. The probe is in
Yeah.
a fair bit under the rock anyway.
What are we at there?
2.
Yeah.
1. Not terribly exciting.
Not very exciting. To the lower left of your screen, though, you see a female that's really moving her arms, and underneath her you can see her egg capsules.
Well, so it is climbing there.
Yep.
Oh. Yeah. To what? Almost 2.5.
That's 3.1,
Oh.
3.2.
Where is he looking?
He's looking up there.
Oh.
I think
Yeah.
there's a lag in my readout anyway. I'm going to refresh my screen.
You know what? I think you can.
Are you guys looking at the gray graphs that you see downstairs in the?
No.
No.
Okay.
Justin set us up with this
Okay.
real-time colorful graphs.
But I'm not seeing that reading, though.
Does it say 4?
Oh, there, now 4.5. Okay.
Okay.
Yeah, let's just sit here for a second, or
Roger.
60 of them.
Oh, sure. We can handle 60. There's a little shimmer in the lower left of the screen that I can pick up.
Oh yeah, like right in kind of this general
Yeah.
quadrant of the computers of the TV screen. Yeah.
Yeah, over these two octopus, it's definitely shimmering. You can see both of the octopus in the lower left of our screen have moved enough that we can see their egg capsules under them.
5.
So now our temperature has gone up to 5.6. And remember, surrounding water is about 1.6. So that's 4 degrees Celsius warmer where the temperature probe is right now.
And
I should also mention, just right above Hercules' arm in the temperature probe, you see something waving, and that is a carnivorous sponge. I think Amanda would want us to tell everyone to notice that. Actually, 2 carnivorous sponges. 3. You keep looking, you see more of them.
they are the strands of hair that look like they're attached to the rock.
That's right.
Correct.
Yes. They're kind of a creamy color. Ropes that are whipping around. Yeah, you'll see a few more if you keep looking.
So this is a neat site. We're up to 6 degrees, and what's also neat is that we have a temperature oxygen probe, a second temperature oxygen probe on the basket, so it's just resting on the front of the vehicle right now, and that's actually picking up an increase in the temperature in the general vicinity of the sub and a decrease in oxygen. Just slighter. It's much more subtle. It's only about 1 degree warmer than the background, but it just shows the diffusivity of this fluid and how it is influencing the kind of the general vicinity of the discharge sites.
Okay, this thing
It's
appears
kinda-
to have maxed out
yeah.
at 6.
Could we
Okay,
then
great.
now try it again, but down in the lower left?
Sure, yep.
Where we can see right now.
You mean by where the octopuses are?
Yeah, to the left of them, I'd say. And maybe we can pan over a little video.
Okay, come a little bit wide video.
Okay.
You mean like between these two and the anemone?
Yes, in that area.
Okay, zoom in a little. It's good there for now.
Is there anywhere particular?
I would say right in that hole, just to the right of the octopus's two suckers.
Oh, over to the right of this guy?
Yeah, I have a strong belief that they like to sit in the best places.
So, like right around here?
Oh, sorry, to the left of her.
Just
It's not my
kidding.
strong point.
Direction.
Zoom in
Yeah,
a little
left and
more
right.
video.
So you want it just a little closer to her, is that what you're saying?
Yeah, I mean, almost if you can just slide the probe into that dark, ominous hole.
Yeah. Yes, exactly. There we go. Okay.
Oh, that tickles.
Hopefully she doesn't grab it. Oh,
So we're
don't
getting a
grab
lot
it.
of
Okay,
really
come wide on the
great
video.
questions here. We've also got a lot of really cool science going on. So if I'm not getting your
Okay, you can do a
questions,
partial zoom.
I really apologize. We do have our
That's good, yeah.
website. A lot of these questions you guys can find answers to, and really great other information by visiting the website,
zoom
nautiluslive.org.
in more.
Obviously, you're there. And questions about the seeps themselves, where we are currently located,
Not getting
all can be found by
Okay.
touring the website. I'll get to as many as I can here as well. It's really cool. We're taking the temperature of the Earth right now here at the Davidson Seamount.
And it might have a slight fever here. We're trying to determine.
Seeing if it's going to respond so far,
So far we don't get a temperature change.
which is a little surprising because it did look promising based
Yeah,
on its shimmer.
Maybe right below her, in that other dark crack.
Oh, there it goes. Start to go up.
Oh,
But it very slowly, so
Okay.
I don't think that's...
yeah. And Dane, you were up when they were doing those other temperature measures. How far into these cracks were they finding those?
They
more?
would go in pretty far.
Uh-huh.
Yep. To my eye, it looked like as far as they could go.
Yeah. Yeah, these ones are pretty much up against hard stuff here.
Okay.
Okay. Let's,
But I should say that there was a lot more shimmer in the water
Uh-huh. Okay.
at those locations.
Yeah, I couldn't really get it to go in anywhere there. It's just
Okay.
up against the rock.
you want to keep looking?
Yeah, let's zoom out.
Okay, full wide, please. Oh my, look at this mess.
Oh.
Well, this is going to cause a problem.
Sorry, are there any metals or materials that octopuses don't like the feel of, the texture of, like copper, for instance?
I bet an aquarium scientist who works with octopuses in captivity would be a person who would
I've
have
heard
much more insight to that.
I've heard AstroTurf works really well, but I don't think we have any of that with us.
I wonder if they wouldn't like the feel of sandpaper or like a
Actually,
gridded surface.
as the director of the Research Experience and Education Facility at UCSB's
yes.
Marine Science
Fill
Institute,
us
I do a lot of
in.
collections myself, and the two-spotted octopus likes to live in holes.
Yeah.
So one of the things that I discovered is my dive knife, which is stainless steel. The end is blunt, I'll gently slip it into the hole, and the octopus will actually grab onto it, and I slowly pull it out, and then I put it in my collection bag and take it to the
Guys,
aquarium
mind
to
if I
share.
try this one?
Yeah,
Nope.
go for it.
Just trying to stretch this cable out, see if this
Yep.
fellow will go.
So then would you interpret that as
Zoom in there,
liking stainless steel?
Yeah, could go either way, couldn't
Yep. Yeah, it could. Like, ooh, get out of here. So the other really amazing thing we saw on the previous 4-hour slot was a brooding female octopus taking in one of the whelk shells that had a snail in it, and she appeared to be enveloping it, taking it down to her mouth, and it all but disappeared. Then her arms opened up again, and when they were all the way opened up and the whelk was laying there over her mouth, her siphon came over and blew it away.
Did you guys confirm that she had eggs?
I don't know that. I don't remember.
Not much going on here.
No, not much going on. Maybe we should look elsewhere.
Yeah.
Yep.
Do you want me to continue logging
please.
these temperatures? Okay.
Guess it's worth knowing that they're not
Yeah.
warm.
At this point, yeah.
So our front porch octopus is now
Now on our temperature probe.
on the temperature probe.
It's like a little hammock.
Well, now we can use the other one.
They're both
Playing on the monkey bars.
A lot of documentation that octopus are very bright and very curious.
Yep.
Do like to play with things, so
Maybe don't make great decisions in life.
they're going places.
Yeah, they're going where the food is.
Well, all right.
And can you talk a little bit about the boxes that are on the left that we used for sampling a bit more?
Yeah, so that box is full of 6 individual fluid samplers that look a lot, that work a lot like syringes. And so the top half that you see, the white squished between those 2 pieces of stainless steel metal, those are actually, those are probably not stainless steel. Don't quote me on the material. Those are the triggers, and so the manipulator arm will wrap his jaws or his claws around that white part, and when he squeezes it, it triggers a spring that you can't see, but it's inside the system, and that causes a syringe to pull up, and the syringe is in the bottom part that's cleared. You can kind of see a reflection of the light, so it's hard to tell right now, but those ones in the general vicinity are we've already triggered them and we've already collected fluid sample. And what we use those for is if we see a place that we want to collect a sample of the fluid in situ and bring it up to the surface and analyze it kind of in near after the cruise as opposed to next year as some of these other samplers we have do, then we'll use a squeezer sampler.
And what are some of the things that you guys are sampling for?
These samples are going to 2 different groups of scientists. One of them is a group of geochemists, and they're interested in the types of elements that are in the fluid, in the water that we're collecting. And then the other scientist is a biologist, a microbiologist, and they're going to be trying to look at what they can learn about the life that might be in these fluids that are discharging from this site.
Thank you.
Right. Can we zoom out, just kind of look around?
Ribbon.
Oh, that was...
So is this at the garden?
This is near the garden.
Okay.
The garden is where we started, and we were having trouble being able to reach where we wanted to there. So we saw these kind of cracks and fissures behind it that we wanted to go and see whether or not we could find an area with more distinct flow
Right.
that we might be able to get to more easily.
Do we want to see where we are with respect to the transit line? Look at this canyon, is just great.
We're right
Can you see
almost
it?
on it.
This is a cool Argus view here.
Outstanding.
The transect line is going to go right kind of past where we are.
That's
Wow,
That's a
look at
really
that.
awesome canyon.
Yeah,
Initial
that is.
shot.
So
Yeah.
like
Look
if
at
we
that.
could get down in there
that's so
at the
cool.
bottom where there's a lot of octopus. At least could we maybe try at least get a zoom in there? I can't hear
Yeah,
you.
we
not
can
us,
hear
Bill.
you.
Yeah, I can get you a look, but I saw when we were coming up, I don't think we have any hope of being able to get in close enough to
Okay.
probe anything.
Just curious to see whether or not we're seeing shimmering going up through where they're kind of in a line.
Yeah, if we keep going to the left when we were out on the,
Yeah, I saw there's a row going up the hill. They're like pretty exposed there.
yeah.
Okay.
Zoom in video.
This is just the way that they're situated as we go to the left and down. They reminded me of the ones we saw this morning. Look,
Is that's shimmering, right?
it's shimmering
Yeah.
going up through that crack.
Yeah, on the left.
It's not a whole lot though. It's not very vigorous.
Yeah.
It's not a whole lot. is it?
Really? To
Well,
the
down
left
at the
there?
bottom.
Yeah, keep
down at the
panning
bottom,
down.
That's really vigorous.
So one of the challenges with trying to collect the in-situ temperature and oxygen data is that we have to make sure that the ROV is able to sit in a safe place
Alright, pump
to
secured.
use certain probes. And so just because we want to get certain data in some areas doesn't mean we can actually do it.
Yeah. I think I see some vigorous flow going up through this area. At least it looks that way to me. But we can't reach it.
Nope.
So like trying to get your hand in the pickle jar.
You got pickles up there?
Sea cucumbers.
Where's an octopus when we need him?
You can see them. They're hiding in that
Get
crevice.
his arms in the pickle jar.
That's true.
Now we're getting down to the bottom of this.
He could screw the lid off the pickle jar
That's right.
and get the pickles from the pickle jar.
That's right.
Yeah, so it looks like there's a decent amount of shimmer back in there.
Yeah, there
Yeah.
is.
Yeah, way in that hole.
Yeah.
If you look at us from Argus's view.
Argus? Uh-huh.
Argus. It's his nickname,
Arc? Uh-huh. Uh-huh.
It looks like we're kind of over a gap. I don't know if we're going to be able to.
What's under us?
Yeah.
I got a question for Jess or Mike, if you can answer this right now. We're not interrupting you guys.
Sure.
Dan Swift from Greeley, Colorado would like to know if there are plans to come back to this site, if you guys would design different pieces of equipment to help navigate some of these challenges that we find this time.
That's a hard one for us to say because kind of the cruise plans, and that's all dependent on our expedition leads, and it's above our jurisdiction. Is that the right word for it?
Pay grade.
Wait.
Our pay grade too.
Yeah. Yeah.
Ooh, there's some nice
There's
shimmer
some nice
in the lower
shimmering
left.
to the lower left.
Yep.
Are we anywhere where we might be able to set down safely?
Yeah, I think we can set down here. This is kind of where we started, I think.
But further back
Yeah.
towards the wall?
Yeah, I think that's. I'm seeing what you're seeing, but that's not a whole lot. I don't think. Full wide. Try and get in there.
Oh yeah, this
somewhere.
is where we started.
Yeah.
Okay.
I don't know how we're going to set down here. Oh yeah, that was where I asked you whether or not you could perch right there.
Cool.
And back where we started. Well, we can keep going to look at what you thought you saw over to the left. If people don't think there's enough shimmer going on here, that is.
Yeah, I would say let's do that, and then
Keep
because
moving.
that brings us towards our transit, and
Uh-huh.
I mean we don't
Yeah.
even know what we're gonna see on the transit.
That's right.
Exciting in itself.
With
Okay.
this, if we are starting to think about those transects, is it important that we start at an end, or can we pick it up mid-transect?
We were talking about that when we did the watch change. We think we should pick it up at the end.
Okay, so that will take quite a bit of stepping to get off to there.
It will?
Yeah, I'll just get an estimate, which is fine. I'm just letting you know.
Yeah,
Pump us
it'd be
back
great
on.
to know how long it would take us to do that.
Yep.
So we're a little bit over 4 hours of pumping for POP. measurements, and
run that
we
Yeah.
estimate
Sorry?
that
Okay,
we've
I'll pause
collected
again.
Yeah, I just turned it on.
145 liters of the 400 that we need.
Say that again, Dane.
Sorry,
How
Dane.
many?
Estimate, well, it would have been 144 at 1952, or 1950 rather. So we're 3 minutes over that. So we've got probably 145 liters.
And how many
Wow.
total?
We want 400.
Okay.
Are we all the way zoomed out on the
Yeah.
camera?
Yeah, you want to come
Thanks.
back in or stay
No,
wide?
I like the out view is nice for trying to screen on where.
What are you thinking about this line of them going up into the left?
I'm looking at that, and then actually over on the wall, it looks like there's some more similar
Yeah,
clustering and lines.
there you go. Yep. Okay, yeah, right.
Again, we're in the Monterey Bay National Marine Sanctuary, about 50 miles off the coast of Monterey,
No.
over the Davidson Seamount, exploring the octopus's garden. Very little is known about these octopus. We
Maybe it's
believe
screening.
they are Mus octopus robustus.
lot of whelks
And
right
we've
there.
seen a lot of other organisms in these areas, sea cucumbers, various species of sea stars, squat lobsters,
Can we push in with video, see how
deep
much
sea corals.
shimmer there is along this line of octopus?
Yeah, go ahead, video.
Not much.
Anemones.
There's a little.
Just a little.
Whelks, which
Wait,
are a
what-
type
they're
of
right
sea
in there.
snail.
Where are those whelks?
Where are they?
Yeah, didn't we see a whole bunch
Oh,
of
they're a
whelks?
little further over, but I was looking, see this little grouping right here? There's a lot of shimmer going across those two octopus.
Oh yeah, right in
Straight to the
behind
lower
them.
left. Yeah, what if we
Mhm.
could get in?
Yeah, yeah, that might be a good spot.
Right by the
Why?
anemone.
Yeah. Right below the anemone.
Look at that.
The red sea
Cranberry.
cucumber right
Sea
there.
cucumber.
Cranberry.
Now you see them, you think of cranberries,
I-
huh?
yeah,
'Cause
that
of
was
what
you.
we-
Jane, you
Yeah.
want me to secure the pump again because of the
Jar
amount of
of cranberry
particulates in the
jelly.
water?
No, that's just.
That's all right?
Yeah, this is okay.
right.
Thanks.
Okay, does anybody else see the hood of the octopus in that rock that we're looking straight at? The eyes on the right and left with the whelks sitting on it.
What?
No.
See the big rock right in front of the
I see
ports.
what you're talking about. He's saying this big rock here that have the whelks on it.
Oh, wow.
Its shape
Right?
looks like that of an. It's the kraken. It's the kraken rock.
That's cool. This is the octopus
There are many
garden.
cracks in the rock.
You could make a song like Crocodile Rock for Kraken Rock. There's some people on board who make up songs with lyrics that are exploration lyrics for popular songs. Maybe we can inspire them to do a Kraken Rock song.
Yes.
Amanda, if you're listening, get working.
A lot of our listeners are quipping in with noticing that the B-52 squat lobster. Somebody mentioned the cranberries.
It was a rock octo.
Go, Amanda. Go, Amanda.
Raiders Online, please send in lyrics. Change your lyrics to match our dive. This would be fantastic.
Can they hear them?
I
Yeah.
heard her. I think she was an SPL.
Yeah.
Oh, oh good, because earlier she couldn't figure out how to be on SPL, so I was just checking. I knew I could hear her.
Oops.
All right. You guys going to write us a song?
Oh, us? Oh, yes, we're working on it.
Okay, perfect. All right.
We need a word that rhymes with whelk.
Elk.
See a lot of sea elks lately, Jennifer?
I used to work in Elkhorn Slough.
Elkhorn is not far
No.
from Acorn.
That's proximal to our location.
Not too far away. Amanda, you want to tell us about a couple of the sponges we see while we're getting the temperature probe here?
Actually, while I do always love to talk about sponges, I looked up a couple interesting things about moose octopus.
Okay.
So I wanted to know why they're called moose octopus, and it turns out that their name, the genus moose octopus, is named after Bent Moose, who is a German researcher who described another species of octopus. Bent Moose. And then also, I looked up a little bit more information about the males with their hectocotylus, that specialized third arm. And it turns out that the way that the hectocotylus works is it has let me check up the word really quick. A spermatophoric duct which carries the sperm packets from the head of the animal, or, well, not the head, but the viscera of the animal where the gonads are, down the arm to a pseudophallus at the end. So that's what that pad is at the end, is called a pseudophallus, and it's that which holds...
We can use the other probe that's octopus free right now.
Yeah, sure.
It's that which holds the sperm, and eventually will put that sperm packet up under the mantle of the female.
All right.
Very cool. Yeah, if any of you think that it sounds like it would be fun to have Amanda as a zoology professor, head on down to Moss Landing Marine Laboratories.
Zoom in there, video.
Great for cocktail parties as well. Her passion, enthusiasm, and knowledge.
Actually, not a bit.
Steve, thank you, because that's pretty much what I talk about at parties,
Hercules.
is fun facts about invertebrates.
But nobody is saying boring.
When I was in graduate school, we would host Halloween parties, and it's really fun to see what costumes graduate students in marine sciences come up with.
Well, even more fun to see what costumes professors come up with.
There were some good costumes on this cruise last year. They had Halloween.
That's right.
On board the ship.
That's right.
I saw a couple anglerfish.
Yep.
I forgot you guys were up for Halloween. That was fun.
It was.
Unfortunately, I was not. I was already.
Oh, that's
had already
right. Yeah,
left. I missed
out.
out on, but I did get some photos sent to me of all the great costumes.
At UCSB, the undergrads can get extra points in the invert class for dressing up as their favorite invertebrate
That's
for
great.
Halloween.
That's great. I love
That
it.
is a great idea.
And with the number of marine invertebrates, you have a lot to choose from.
Oh, you want to go in pretty much right under where I have it, between the two octopuses?
Yeah, that would be great. On the bubble cam?
That?
See on the bubble cam?
Yeah.
That big
Yeah.
hole?
It's the whole.
Yeah.
Okay. Yeah, it's the same place I'm thinking.
Okay, come on. Good readings. No whammies. I think I dated myself with that reference.
Yeah. Nobody laughed.
I'm
What's
like, what's
wrong?
a whammy?
It's an old game show. But I heard it's coming back. So everything that's old is new again. I forget what it was called.
So science, it's the graphs in the center then, right? Oxygen concentration,
Yep.
saturation, and temp. That's all from your sensor, yeah?
Yep.
So far, I don't have any changes.
And is the Mesotech screen showing temperature of that probe?
No, that's
Okay.
just our probe.
Got it. Okay.
But if you go to, if you could see the Chief Science's computer, they're looking at the oxygen probe now.
Are we
Okay.
getting it?
I'm
getting
not
anything
really
on it?
seeing any change at all.
I'm not
No.
seeing any change.
But
What
we are
about-
seeing it.
Is it moving at time?
It's not, but none of the graphs are updating. Oh, I have a 5-second delay. Let me change that.
Somebody just wrote in, try tic-tac-dough. Where the whammy came from.
Oh.
Oh, somebody else
And-
said Joker's wild. Well,
I thought it was something like pressure luck, but
I'm not seeing any
Science.
change right here.
Science, can you, looks like they're in the upper right-hand corner. There's an update every last 1 hour. Can you reduce that to something that's a little more timely?
Oh, this is, can
think
we
that's
please
just
reset
showing
the
the last
oxygen
hour,
temperature
wasn't
probe?
oh yeah, we're getting nothing.
It was, but
Yeah, so we're getting,
Oh, it's just
this
scroll.
one's...
we're not
it
getting
was
any data right
today.
now. Thank you for noticing that.
So Jess, is there a way for them to reset the update time?
Not not sure. It looks like it's saying it's supposed to be refreshing every 1 second, but that's not necessarily
Can you
happening.
actually turn, I know they've had to turn the probe on and off.
Was that this
a few
probe that they
times.
Yeah,
The
started?
incision one, yeah.
Yeah, I
Can we do a
can,
reset on that?
I can reset that.
Thank you.
All right, off.
Multiple confirmations that it was press your luck.
Oh, yes.
Press your luck is the correct answer based on the
All right, back on.
World Wide Web.
Thank you.
Thank you guys out there.
Give it a second, see
Yeah,
if it
So
shows up.
we had to
definitely
make
see
a phone
shimmering
call.
here.
Uh-oh.
What?
We had to make a phone call?
Yeah, we had to make a phone call to get the
okay,
answer.
this is great. This is good. We're at 8.8.
Oh,
Nice.
yeah, okay.
Thank you for noticing that, Jessica.
Yeah, so,
Nice.
and what we were hearing from the watch before ours was that when they were using this probe, where the temperature probe can't be put as far into the rock as with Herc's probe, they were seeing that the measurements were a few degrees lower.
Does that mean we should get Herc's probe?
Maybe we want to do that
Stick
next,
it
but let's
into the
get
rock.
but let's get a little bit of more readings with this one first, I think.
While we're sitting here watching that, we are 300 meters off the end of that line.
Okay.
So take at 0.2 knots, which would take a bit to get up to, probably be about an hour, 50 minutes to an hour.
Oh, really?
Depends on how much speed we pick up and how comfortable the pilots are as far as speed.
Can we, could we follow the line down, at least make that hour a
Yeah,
Sure.
part of a
I
transect?
think it would make sense to just pick it up here. Can
9.5, not bad.
No, it's good.
So 9.5 with this probe, it means if
That
we get
means we win,
Herc's
doesn't
probe,
it?
we might win.
No,
Yeah.
not
Most
bad.
importantly,
Wow.
We
do
got to
we
meet
win yet?
10.4.
I know, but,
Well,
like,
but
we
at
haven't
different
done
probes.
Herc's probe yet.
Yeah.
The 10.4 is with Herc's probe,
Oh,
so
yeah,
there's
well,
a
we already
chance
won.
here, people.
And we got 9.5 with this probe at a previous site, so we may have already
Ooh.
beaten them today.
Yeah,
Look at
no
that.
problem. 3
I
hours
mean,
left.
We're all friends here.
Not
but
that science
doesn't
is a
mean we-
competition.
Yeah, but it's still fun to win.
It might be from the O2.
I'm
going
filling
off.
out my
Constrictive
bracket.
messages.
Maybe.
One of the things that's so amazing about deep sea exploration is so little of it has been done, and there's so little that we actually know. We have a question out here about how many eggs does the average octopus lay, and I'm going to assume that we're talking about the moose octopus here, but do we even know the average number that they lay yet?
20
They're
to
conferring.
35. We were conferring. On average, like 20 to 35, but they can be more than that in a clutch as well. Okay, well, our numbers seem to be kind of stabilizing with an oxygen saturation around 60
Mhm.
and temperature around 9.6. I wonder if we could try one other location in this area. So,
Sure.
but remember this one, because this might be where we want to replace with Herc's probe.
Yep.
Okay, thank you.
Now we're limited in reach.
I
Just
was
up
thinking-
the crack here where the lasers are. I could get in there.
Well, there's either up there, or I was looking at the bubble cam, and there seems to be a hole further towards Herc.
That's like 9.8.
Closer towards Hercules?
Yeah, well, I'm looking at the bubble cam,
Yep.
and it looks like the probe is kind of going out at about 45 degrees, and I was thinking straight down, there's a hole there as well, or we can go out.
Yeah, you mean in the same frame?
Yes, in the
Yeah,
same
yeah.
frame, yeah.
Yep. We're right here. We can do that one.
Because there's a couple octopus way down in there, and I'm just wondering if... Okay, so that measurement was right below that anemone. Now we're just going to look just a little further down.
Someone asked 60
It's just
what
closed up
for
now.
oxygen?
Percent saturation.
Percent saturation, thank you.
Yep.
Just we've got a one of us out there right now. You guys make me so
Oh,
excited. I'm
no,
pursuing
it's
a
okay.
Bachelor
Yeah.
of Science to
I
become
should,
a
though.
marine biologist.
welcome aboard.
They cannot wait to join the field and start discovering things.
Going
Well,
up at all, Jess?
apply for a
Looks like it's around 9.
internship on the Nautilus. You can be out here with us.
So far this one's
No,
a little
dropping.
lower in temperature. This doesn't look to be as
8.
good of a location. Maybe we should try that. Could you reach that one above
Yeah.
the other octopus? Plus, I don't really want her to hold on to us here.
Looks like the oxygen's higher here than the
Yeah.
other site.
Yep. We might not be able to get far enough in because of all the octopus that are there.
You want to try and go in here?
Yes.
I think this is
Ooh.
a good spot.
Yeah.
That's an awesome spot.
So I was thinking about something else. Were you asking about what the oxygen measurements were?
Mm-hmm.
So we're looking both at millimoles per liter and we're looking at oxygen saturation.
5.
So when I was saying 60, that was millimoles per liter.
Ah.
Thank you for that clarification.
So millimoles per liter is concentration, and
5.
percent saturation is
5.8.
percent of the maximum amount of oxygen that could be
See if it
contained in the
updates
water
different.
of that temperature.
So right now, we're not getting very good measurements.
Is that reading 5.3?
5.3.
That's low.
Yeah.
So it looked like a good spot, but you
Yeah.
know, looks like the first spot was the best one. Right below that anemone.
Is there an inverse relationship between oxygen saturation and temperature?
Yes.
Yep.
Sorry.
Quite linear.
What about over to the right between the,
Between those other two octopus,
Yeah.
where it's shimmering and there's a shrimp.
yeah.
Yeah. Just take a quick.
I would imagine
there.
depth plays a role in that as well.
Well, there is a relationship with dissolved solids, so salinity. So to the extent that deeper waters are more salty, then there is a relationship.
Interesting.
So far, we're not seeing the temperature rise above 5.3, no 6 over there.
No.
No.
I think the first spot was the best one.
Yeah.
Mm-hmm.
Let's see if we can get out the Herc probe.
Yeah. And then if, this might be an appropriate place to deploy that second temperature logger.
Yeah.
Or the fourth temperature logger, actually.
So my question is whether we can all remember where the first one was.
The first temperature logger?
No, where?
Oh, it's below that anemone.
Got it.
So the kind of mustard colored.
It's like a peach pit.
It does kind of look like a peach pit. That is
Right.
a closed up anemone, and it
Okay.
was right
Yep.
under
Yep.
that.
Okay.
Come wide on the Zeus, please. No.
So Aaron, it looks like we're just a little, northwest of the octopus garden. Is that right?
Yeah, northwest of the location is marked.
That ledge
Um,
that we call the octopus garden?
so that was the initial octopus garden. We're only 5 meters off that.
Okay.
Maybe 10 to 15 with error.
Okay.
So we're at about 3,400
Is
meters.
that the octopus on it?
Yeah,
I think it's
that's
holding
octopus.
on.
Yeah, he's holding on.
Let go.
Seeing some amazing things. It's going to be about a 4-hour drive back to the surface when we're done down here.
There we go.
Sometime perhaps tomorrow. That's a really great time to find out more about things like what you're